Hello, and welcome to our discussion of microbiology, looking at how we are able to observe microorganisms. One of the tools that we use the most in our study of microbiology is the microscope. And if we look at the word microscope, we're looking at micro meaning small, scope meaning viewing or looking. So we are using our microscopes to view these organisms that are not able to be seen with the naked eye. Now, in order to understand how microscopes work, the first thing that we have to look at are the properties of light. So in section 2.1 in your OpenStax textbook, you have this introduction to the properties of light. And when we look at light, and in particular visible light, because that's what we're concerned with, it contains waves that behave like other electromagnetic waves. So light behaves, or visible light behaves in the same way as X-rays or radio waves or television waves. If we look at waves, or a wave here, as you see in figure A, you see a couple of different properties. We have the amplitude and the wavelength and frequency. So the wavelength is the distance between peak to peak, okay, or trough to trough, okay. So that is the wavelength. The amplitude is the height, okay, of each peak or the depth of each trough, okay, you see here. And the frequency is the rate of vibration or the number of wavelengths within a specified time period. So we see when waves are not coming as frequently, then that would be a low frequency. When the waves are closer together, that would be a high frequency. And it's this idea of wavelength and frequency that allows us to discern visible light from other forms of electromagnetic radiation. Now, the next section gets into some definitions in terms of how light behaves. So, as light comes in, it can be reflected, it can be absorbed, or it can be transmitted. So, reflection is when light comes in and it bounces off of a material. And when we see color, this is seeing these reflections. So if we see something that is red, essentially what is happening is that the red light, okay, so that portion of the visible light spectrum that is red is what is being reflected and bouncing off of that substance into our eyes, and the other colors are being absorbed. And absorbance is when whatever material we're looking at captures that energy. So essentially reflection and absorbance are opposites when we're thinking about light. Reflection is when the light waves bounce off, and absorbance is when they are absorbed. They actually go into the substance. And transmission is when the light actually travels through the material. You can kind of think of transmission like light going through a glass. Okay. When a material, when a substance allows a lot of light to be transmitted, then we say that it's transparent or has more transparency. If it prevents the light from being transmitted, then we say it has opacity or it is opaque. Okay. Light waves can interfere with one another as well, and that's called interference. And then diffraction is when the light is bent. So all of these different interactions of light can wreak havoc, really, with what we're trying to do using a microscope. And so microscopes are built so that we can reflect the image that we need to see back to our eyes while reducing or attempting to reduce diffraction as much as possible. So if we look at lenses, so if we think about microscopes as a series of lenses, so we have to consider refraction. This is when light changes direction. So you've got this image here, this image A. You've got light coming in. It hits this piece of glass or this transparent material, and the light is bent. Okay, it is refracted. It is changing directions. And depending on what this material is, we will see different levels of refraction and different speeds of refraction. Light moves more slowly through water than it does through air. So when light enters water, it has a high refractive index compared to air. And here's the image of this. This is a pole that has been put into the water, and you see that it appears bent. And this is because of that refractive index. That the refractive index of water is much higher than air, so we kind of get this optical illusion. We get this idea that this rod or this pole is bent when we know that it's not. So we have to be careful when we're looking at items or looking at living organisms, or when we're looking at preserved organisms through the microscope, because we want to reduce this refraction as much as possible. So this next series you see here of images, we've got a prism, we've got a convex lens, and we have a concave lens. And you can see that we get different refractions with each one of those. And the idea here is when we're looking at this refraction is that we want to have some sort of focal point. Okay? The focal point is the image point when light entering the lens is parallel, because you have to consider that whatever we're looking at, we're going to have this focal point. And it's the same thing with, with our eyes. We have a focal point. We have this area within our eyes, the retina, where images are going to be focused. And so when we're using microscopes, we want to make sure that that focal point is where it is supposed to be. So we can manipulate our microscope. We can manipulate what we're trying to view by controlling the distances. We have the ability to move on our microscope. We can move the stage closer or further away from the lenses. We can use different types of lenses with different magnifications. And in fact, the microscopes that we use are called compound microscopes because they use more than one lens in order for us to view these specimens. And then we look at the curvature of the lens, whether we're looking at a convex lens or a concave lens. So microscopes are built with this idea in mind, knowing that we are going to have to overcome this refraction and the diffraction and reflection, that we're going to have to overcome these different things. Microscopes are built for us to best be able to overcome those issues and view the specimen as it really would be. Now, visible light is part of the electromagnetic spectrum, kind of stuck right in between ultraviolet and infrared light. And so when we are using microscopes, again, like I mentioned, we're using light microscopes, and we're referring to this visible light. But sometimes we have to use things that are a little bit different. There are different types of microscopes that we can use to essentially use some of these other forms of electromagnetic radiation, but all we have are these simple compound microscopes that are going to use visible light. Okay. Now, something else that we consider when we're using microscopes is the magnification, which is the ability of the lens to enlarge an object. So that's the whole goal, right? That we can't see these things with the naked eye. So magnification is the ability of that lens to enlarge an object. Resolution is to be able to tell that two objects are separate. In other words, resolution is really the sharpness of the image. You want to have high resolution to be able to distinguish between different parts of an object. If you have poor resolution, then your image is going to be fuzzy. Okay, and our wavelength affects resolution, and then the aperture affects resolution. Aperture is the ability of the lens to capture light. Remember, this all comes back to light. And if we have a lens that can capture light a little bit better, then it's going to improve the aperture, which is going to improve resolution. And then the third thing that we consider is contrast. Contrast is when we want to have a difference in appearance, typically between what we're trying to observe and the background. So we use a lot of different stains and dyes to increase that contrast. When we have something that has been stained, it's going to appear darker against a lighter background, and that allows us to be able to see that organism better. Sometimes we can increase the contrast by staining the background and not staining the organism itself, and that's typically called negative staining because we're staining the area around what we're trying to see. And we'll get more into that contrast and the idea of staining when we actually get into the lab a little bit later on. Now, if we take a look at the history of microscopes, we have several individuals who were important in this development of microscopes, and von Leeuwenhoek is number one. He is sometimes referred to as the father of microbiology because he invented the first microscopes that allowed us to be able to view this world that was previously unknown. We had this idea that maybe there were things out there that we couldn't see, but we weren't aware of them, we couldn't observe them until Leeuwenhoek came along and built his microscopes. I'm not going to talk much about these because in the presentations that you're going to do for Unit 1, you'll learn more about von Leeuwenhoek and Robert Hooke, who really were instrumental in this field of microscopy. Now, when we look at microscopes, we have a lot of different types of microscopes that we can use. In our labs, of course, we are using light microscopes, and specifically we're using bright field microscopes where we have a dark image on a bright background. They are binocular, which means they have two eyepieces that we look through, and we can get a total magnification of a little more than 1,000 times before we start to lose the resolution, before we start to lose the sharpness of our specimen, what we're trying to observe. And we'll look more specifically at the parts of a microscope when we get into the lab. Dark field microscopy has modifications to the condenser so that it doesn't, this disc essentially blocks a lot of the light coming through. We have phase contrast microscopes, and we have DIC microscopes, we have fluorescent microscopes, and we have electron microscopes. I'll let you read about those on your own.